All right, here are your cell respiration video notes. And these notes are going to cover a few processes. So they're going to cover glycolysis, the Krebs cycle, electron transport chain, and fermentation. So who even does cell respiration? The answer is animals, although plants can do it too. We take in oxygen, or O2, and what do we release? CO2. So we are heterotrophs, meaning we have to consume something else, right? That something else is food. Then the answer becomes, where does our glucose even come from? Our food. And here's a cute little picture of a cow. So the glucose is going into the cow, into the cells, and the mitochondria is going to make energy out of that glucose. So when we look at glucose in general, so it's in our food, and our cells take the electrons from glucose and they break it down. This, as a result, generates ATP. And if we remember, ATP is a source of energy. And here's just a look at the structure of glucose. So what about metabolism? So if we remember, metabolism is when you break down molecules to provide energy. In this case, we're taking glucose and we're making ATP, more specifically, catabolism. But do we use all the glucose we produce? Do we use all the glucose we eat, I should say, at one time, or ATP we produce? No. So how often do we eat? We're not eating every second of the day. We can eat meals and still be able to process that glucose slowly to produce ATP. So here's an overview of cell respiration in general. So we start here with glycolysis, and then from there it's going to either do one of two pathways depending on whether oxygen is present or not. So the formula for cell respiration is the opposite of photosynthesis. So we're taking in glucose, taking in oxygen, we're releasing CO2 and water, and as a result, we're getting 38 ATP per molecule of glucose. So what do you notice? Like we said, it's the opposite of photosynthesis. And what's the purpose? Create energy. We eat to create energy for our cells because we can't make our own food. So another term you need to be familiar with is oxidation and reduction. So oxidation is known as the loss of electrons, or it can also be the removal of a hydrogen ion. And reduction is the gain of electrons or the addition of a hydrogen ion. The easiest way that I use to remember this is oxidation starts with an O and there's an O in loss. So if you keep oxidation and loss together, then you'll know that reduction and gain go together. So here's the basics of glycolysis. So step one, glucose is going to enter the cell. This is from our food source. We need 2 ATP to even start this reaction. So once we've started the reaction, glucose is going to split into 2 separate 3-carbon compounds. They're called pyruvic acid or pyruvate, and as a result, we have 4 ATP released. We also release 2 NADH, which will come in handy later. But it's really important to note that here our net gain is only 2 ATPs. And you may be wondering, well, why? We made 4. But think about it. You needed 2 to start it. So if, let's say, you're putting change into a change machine, if you put in 2 quarters and you get back $1, how much did you really get? 2 quarters. So that net gain is 2 ATPs. So here's a look of the overview. So we can see our 6-carbon sugar here, glucose, going in. We're getting those ATPs and the NADHs, and as a result, we're getting 2 3-carbons known as pyruvic acid. And just so you're keeping track, so far we have a yield of 2 ATP. So before we go into the Krebs cycle, there's a little bit of prep. So this Krebs cycle prep is only if oxygen is present. In this case, the pyruvate's carried into the mitochondrial matrix. So in this preparation stage, a lot of stuff goes on. First of all, NAD+ is reduced to make more of that NADH that we'll need later. We also release CO2, and something called coenzyme A is going to join with that pyruvate we've made. This forms a really important product known as acetyl-CoA. And please pay special attention to the fact that because we made 2 pyruvates, technically there's 2 acetyl-CoAs that are going to enter the Krebs cycle for each 1 molecule of glucose. So this just shows that once again in a picture form. So you can see the pyruvic acid entering, that CO2 is exiting, coenzyme A is entering, and then we get acetyl-CoA. And like I mentioned, we're also making more of that NADH that will be very important later. Before we go any further, I want to just take a minute to look at the structures of the mitochondria. So there's an outer membrane and an inner membrane, but there's also something called the matrix. So the matrix is all of that space in between the mitochondria, but even more important is something called the cristae. Those are all these little folds you see in the mitochondria. And what that does is that increases the space because there's more areas for these processes to happen. So the Krebs cycle, that coenzyme A that we talked about, it helps that acetyl-CoA enter, but it doesn't continue through the cycle. So we just have acetyl-CoA going, and that's slowly deconstructed. So those electrons are going to get stripped off, and we release more carbon dioxide here for each acetyl that enters. So remember, once again, there's 2 acetyl-CoAs, meaning there's 2 turns of the Krebs cycle. It has to happen 2 times. And each turn yields the following. So we get 1 ATP, 3 NADHs, and 1 FADH2 for a total yield per 1 molecule of glucose. We're getting 2 ATP, 6 NADH, and 2 FADH2s. So here's that overview picture. Once again, it shows the acetyl-CoA entering, but that CoA jumps off. Then we have our CO2, our NADH that's made. Here's that ATP we really need, and that FADH2. So we have a net yield here of 2 ATP, and we're up to 4 molecules of ATP so far. So let's pause, talk a second about electrons. So electrons are super important to this next process. And it's also important to note that electrons move as part of a hydrogen ion. They don't move alone. So the main function of the glycolysis and Krebs cycle, and this is really, really, really important, you may want to highlight this, is to provide electrons for the electron transport chain. So why do we even care about the electron transport chain? Well, so far we've only made 4 ATP, and that's not enough to do your body's life processes. So the electron transport chain is where we make all of the ATP that we need to actually make this worthwhile. So this chain operates by a process called chemiosmosis. And this is just simply the movement of ions down the electrical chemical gradient. Now, osmosis, we've talked about, is the movement of water down the gradient. So this is the movement of ions. All right, so here's the electron transport chain. So there's some basics. First of all, hydrogen ions are going to be released into that intermembrane space. The energy released from the electrons as they move these hydrogens across the membrane into this space is going to result in a gradient with lots of potential energy. Then those hydrogen ions are pumped through ATP synthase into the mitochondrial matrix. And this is where we create all that ATP from ADP. So very, very, very important, this statement here. I would definitely be highlighting that in your notes. And we also need to realize that NADH is releasing hydrogen ions during this process, and that's going to combine with that oxygen that we're breathing in and creates that water, which is another product of this process. So here's a look at the overview. So you see the electron transport chain here, and then ATP synthase is here. So NADH is getting reduced to NAD+. So all those NADHs that we made in those previous processes are coming here, and they're getting reduced to make those electrons move through the electron carriers. So as a result, we're also getting those hydrogen ions that are going to combine with that oxygen there to make water. And then most important, that ATP synthase where all that ATP is made. And if you look here, wow, 34 ATP just in this step. So we've only made 4 prior to this, and this step created 34, which is huge. So our final yield is going to be 38 ATP, and you may see it as 36 in other places, but for the sake of this class and modern science, we're going to go with 38 ATP per 1 glucose molecule. And this just outlines the whole process for you. There are unfortunately some times that the electron transport chain can go wrong. So what happens is that ATP formation is blocked at different steps by toxins. So if you look here, you have rotenone. This is an insecticide. It's not allowed anymore, but prior it did have some issues. Things like cyanide, carbon monoxide, those are going to also prevent that. And then we have oligomycin, which doesn't even let the ATP synthase work. So the other thing we haven't discussed yet is fermentation. So why do we care? Why would we even do fermentation? You're going to do fermentation when there's no oxygen present. And in this case, glycolysis only makes 2 ATP and has a limited amount of NAD+. So we need to figure out a way to get more. Well, it's used during glycolysis, so there isn't a way to make more unless we do fermentation. And the whole point of fermentation is to regenerate NAD+, and kind of as a bonus, we get a little bit of ATP on the side. So remember, fermentation is an anaerobic process, meaning oxygen is not present for this process. So there's 2 types that you need to know. There's lactic acid and alcohol. In our bodies, we cannot do alcohol fermentation. We can, however, do lactic acid. So this is if there's no oxygen present, but the cell needs to regenerate NAD+ and ATP, that pyruvate, instead of going into the Krebs cycle, is going to go into fermentation. So here's a look at alcohol fermentation. So this is when that pyruvic acid is converted into CO2 and ethyl alcohol. And this is something very common in yeast cells. They release that alcoholic waste, but unfortunately they can release too much and sometimes they die. But let's look here at the picture. So the red box is going to summarize the process of fermentation. The left of that is the process of glycolysis, which we've already talked about. So it shows more NAD+ being created, which is what we want, but it also shows that release of CO2. Lactic acid fermentation, on the other hand, is when pyruvic acid is converted into lactic acid. It still creates NAD+, but no CO2 is released here, so it's not the same process. And this happens just when that NADH is oxidized. Remember, oxidized, O for loss, so we're losing electrons and removing hydrogen. It's also used to make cheese and yogurt, but those of you that have ever gotten cramps or a side stitch when you exercise, this is lactic acid buildup in your muscles. And that's all for your notes.